course title is Microbial Technology and the title of lecture is Detoxification of Pollutants under Swami Vivekanand Technical University, Durg. Lastly, we discuss about the topic. There are three different types of horizontal transmission for the transfer of genetic information: conjugation, transduction and transformation. Transduction is the process of the transfer of gene from the recipient to the donor through bacteriophage. Transduction is two types: generalized transduction and specialized transduction. Generalized transduction is the process by which any bacterial gene may be transferred to another bacterium via a bacteriophage, and typically carries only bacterial DNA and no viral DNA. In essence, this is the packaging of bacterial DNA into a viral envelope. Now, a specialized transduction is the process by which a restricted set of bacterial genes are transferred to another bacterium. The genes that get transferred, donor gene, depends on where the phage genome is located on the chromosome. Transduction does not require physical contact between the cell donating the DNA and the cell receiving the DNA, which occurs in the conjugation, and it is DNA's resistance. Transduction happens through either the lytic cycle or the lysogenic cycle. Transduction is especially important because it explains one mechanism by which antibiotic drugs become ineffective due to the transfer of antibiotic resistance genes between the bacteria. Topics to be covered in this lecture: introduction, type of pollutants, types of detoxification by bioremediation ex situ or in situ, mechanism of detoxification, microbes involved in the remediation, factors affecting in detoxification and the advantage, limitations, summary, conclusion and references. Bioremediation refers to the process of using living microorganisms and plants to remove the environmental pollutants or prevent pollution, mass, toxicity, volume, concentration of the pollutants without human intervention. Means living, and remediate means to solve a problem or to bring the sites and affairs into the original state. Bioremediation technology using microorganism was reportedly invented by George M. Robinson. Conventional method of the bioremediation are cap and contents, maintain it in same land and dig up and remove it to land fills. There are two types of conventional method. Types of contaminants: biodegradable contaminants, partial degradable contaminants and non-degradable contaminants. There are three types of contaminants. Biodegradable contaminants means petroleum products such as gas, diesel, fuel, oil, crude oils compound, benzene, toluene, xylene, some pesticide, malathion, some industrial solvent, coal compound, phenol, cyanide and coal, etc. Partial degradable are TCE, trichloroethane, treated to groundwater, PCE, perchloroethane, dry cleaning solvent have been degraded in labs but not in fieldwork, arsenic, chromium, selenium, etc. Not degradable pollutant are uranium, mercury, DDT, etc. In order to break down of transformed harmful substance into the less harmful molecule, bioremediation uses the amazing catabolic diversity of microorganisms which play a significant part in the abstraction of pollutants. Strategies for the bioremediation: organism must have necessary catabolic activity required for degradation of contaminant at fast rate to bring down the concentration of the contaminant. The targeted contaminants must have bioavailability. Third is soil conditions must be favorable for the microbial or plant growth and enzymatic activity. And fourth one is the cost of bioremediation must be less than other technology of the removal of contaminants. Types of bioremediation: there are two types of bioremediation on the basis of site: in situ and ex situ. Further, in situ is divided into two parts, engineered and intrinsic. Then ex situ is divided into the solid phase and slurry phase, which we saw further. Now, in situ bioremediation techniques. This method entails handling contaminated material on the pollution sites, because it does not involve excavation. There is minimal to no alteration of the soil's composition, since there is no additional cost for the digging process involved. This technique should ideally be less expensive than the ex situ bioremediation method. It is superior method to cleaning contaminated environments, since it is cheaper and use harmless microbial organisms to degrade the chemicals. It involves the supply oxygen nutrition by the circulating aqueous solution through naturally occurring bacteria to degrade organic compounds. In situ bioremediation is applied to the degradation of the contaminants in saturated soil and groundwater. However, an important issue is the cost of designing and installing some expensive machinery. Types of in situ bioremediation. There are two types of in situ bioremediation. First is in situ bioremediation and second one is engineered in situ bioremediation. In situ bioremediation, in intrinsic bioremediation, naturally attenuation, also known as intrinsic bioremediation or natural remediation, conversion of the environmental pollutant into the harmless forms through the innate capability of the naturally occurring microbial population, is called intrinsic in situ bioremediation. This is the figure in which we saw in situ intrinsic bioremediation, where directly microbial population which is present in the inside the pollutant site is participated in the bioremediation activities. Now, depends upon the natural processes to degrade the dissipated contaminants in soil and groundwater. Natural attenuated process include physical, chemical and biological transformation. Advantage of the in situ intrinsic bioremediation is contaminants are ultimately transformed to innocuous byproduct, etc. For example, carbon dioxide and water. Most of the time not just transferred to the another phase or location. Within the environment, intrinsic bioremediation is non-intrusive and allows continuing use of infrastructure during the remediation. Intrinsic bioremediation is less costly and intrinsic bioremediation is not subjected to limitation imposed by the use of mechanized remediation equipments. Applications: diverse microbial population indigenous to subsurface environments can degrade important classes of organic contaminants. The extent of intrinsic bioremediation depends on the bioremediable ability of the contaminants and on the site's hydrogeologic and chemical characteristic. Four types of hydrogeologic and geochemical characteristic determines the successfulness of intrinsic bioremediation instead of an engineered cleanup system. Predictability of the groundwater flow and sufficient amount of the electro, etc. Adequate capacity of the buffer against pH change that may occurs with the increased microbial activity, and the site must have a natural supply of the elemental nutrients. When intrinsic in situ not sufficient to degradation, then scientists move to engineered in situ bioremediation. What is engineered in situ bioremediation? It accelerates the degradation process by enhancing the physiochemical conditions to encourage the growth of microorganisms. They further classified into 6 types are following: biosparging, biostimulation, bioaugmentation, bioventing, bioattenuation and bioslurping. Engineered in situ bioremediation. First of all, we talk about bioventing. Through the controlled stimulation of airflow, these strategies promote The activity of native bacteria in unsaturated zone, hence enhancing bioremediation. Achieving microbial transformation of contaminants into a harmless conditions is the ultimately goal of bioventing, which involves amending the bioremediation process by adding moisture and nutrients. This is figure 3, which is showing in situ bioremediation by bioventing. Next one is bioslurping. This technique combines vacuum enhanced pumping, soil vapors extracted and bioventing to achieve soil and groundwater remediation by indirect provision of oxygen and stimulation of contaminants biodegradation. The technique is designed for free product recovery such as light non-aqueous phase liquid LNA-PLS, thus remediating. Figure number 4 is showing in situ bioremediation by bioslurping. The capillary and unsaturated saturated zone etc. involved in this process. It can also be used to remediate soil contaminated with volatile and semi-volatile organic compounds. Next one is biosparging. By indirectly supplying oxygen and promoting the bioremediation of contaminants, this method integrates bioventing extraction of soil vapor and vacuumed enhancing pumping to remediate soil and groundwater. Light non-aqueous phase liquid LNA, PLS and other free product recovery are the focus of this technique, which also addresses capillary, unsaturated and saturated zones. Now, biosparging involves the injection of air under pressure in below the water table to increase groundwater oxygen concentration and enhance the rate of biological degradation of contaminants by naturally occurring bacteria. Biosparging increase the mixing in the saturated zone and thereby increase the contact between soil and groundwater. Figure number 5 is showing in situ bioremediation by biosparging. In easy and low cost of installing small diameter air injection points allow considerable flexibility in the designing and construction of the system for the biosparging. Fourth one is bioaugmentation. Bioaugmentation is the introduction of a group of natural microbial strain or genetically modified engineered variant to treated contaminant soil or water. Figure number 6 is showing in situ bioremediation by bioaugmentation technique. This is a technique which is commonly used in municipal wastewater treatment to restart activated sludge bioreactors, mostly at the site where soil and groundwater are contaminated with chlorinated ethanes such as tetrachloroethylene and trichloroethylene. Bioaugmentation is used to ensure that the in situ microorganism can completely degrade these contaminants to ethylene and chloride, which are non-toxic. Monitoring of this system is difficult. Numerous studies indicate that introduced microorganisms do not survive in the environment and thus do not increase contaminated remediation. Further, number 5th is showing bioattenuation. Attenuation means during the attenuation biodegradation occur naturally with the addition of nutrition and bacteria. The endogenous microorganisms present will determine the metabolic activity and act as a natural attenuation, while there is no anthropogenic involvement in bioattenuation. The contaminated site must be still monitored. Usually, addition of electroacceptors such as oxygen, nitrogen, sulfate, or electron donors such as organic carbon, hydrogen, etc. could involve bioaugmentation, adding the catalyst for bioattenuation. In figure number 6, it is showing. Next one is biostimulation. Biostimulation or stimulated aerobic biodegradation is the practice of eliminating the factor that limited the bioremediation of the pollutants, usually introducing nutrients such as nitrogen and phosphorus and oxygen is in the saturated zone and in the groundwater to increase the number and activity of the autochthonous microorganism capable of biodegradation. By supplying oxygen to the subsurface, biological process are stimulated and aerobic biodegradation rate can be accelerate 10 to 100 times compared to the natural rate. Compound treated by biostimulation most frequently are petroleum hydrocarbon components, BTEX, HAP, TPH and related oxygenated ethers such as MTB, ETB, TBA, etc. Figure number 8 showing in situ bioremediation by bioattenuation of arsenic and iron. Advantage of bioremediation by in situ bioremediation: do not require excavation of contaminated soils. It is less expensive, less chance of contaminated other sites. Possible to treat a large volume of soil and groundwater at once, and no remediation waste are produced because treatment occur below ground. A limited transportation cost and disposal charge are minimum. Land requirement and environmental disturbance are minimal. Take hard to withdraw hydrophobic pollutant that may be located at depth and prohibited excavation. Disadvantage of in situ bioremediation: it is slow process and difficult to manage, require uncompact soil, difficulties to overcome mass transfer through mixing or surfactant addition, therefore slow degradation of hydrophobic pollutants, difficult to implement, highly stratified soil that hinder the vertical distribution of the injected air or gas through the contaminated zone. Ex situ bioremediation: in ex situ bioremediation generally it is selected for the treatment of the highly contaminated material, applicable when contaminants has been extracted from the source zone. Hydrophobic pollutants such as PAHs and PCBs are not effectively removed in situ due to the low availability. Rapidly removal of contaminants is required. The average time for ex situ bioremediation is about 60 to 90 days, hence faster compared to in situ bioremediation. It is better controlled area, depth, nutrition, oxygen, moisture and temperature, etc. In ex situ bioremediation, procedures are typically evaluated by lighting of the following factors. First one is the expenses of treatment, the kind and the depth of the pollutant, the degree of the pollution and the location of the contaminated site is geologically. The performance required that also influence the selection of the ex situ method of the bioremediation. On the basis of contaminant under treatment, ex situ bioremediation is classified into two types. First one is solid phase and second one is slurry phase system. Solid phase system is divided into four parts. First one is land forming. Land forming is a simple technique in which contaminated soil is excavated and spread over a period bed and periodically tilled until pollutants are degraded. Land forming is one of the most straightforward bioremediation method because it requires nothing in the way of equipment and is inexpensive. The drawback include the need for large operating area, a decrease in microbial activity because of the unfavorable environmental conditions, extra cost associated with the extraction and the lower removal efficiency of the organic pollutants. This is the figure which is showing ex situ solid phase system bioremediation by the land forming. Composting. Composting consists of excavation the soil and then mixing organics such as wood, hay, manure and vegetable waste with the contaminated soil. The presence of the organic material support the development of the rich microbial population and elevated temperature characteristic of the composting. This is the figure in which we show ex situ solid phase system bioremediation by composting. Next one is biopiles, typically used for the treatment of the surface contamination with the petroleum hydrocarbon. They are a refined version of the land forming that tend to control physical losses of the contaminants by leaching and volatilization. Nutrient amended are then added and occasionally aeration is used for the improve the bioremediation process by the essentially boosting microbial activity. This method consists of a treatment bed, irrigation, aeration and the system for the collecting leachate and nutrients. Due to its advantageous qualities such as cost effectiveness and the ability to effectively facilitate biodegradation under the right conditions such as temperature, aeration and the nutrient management, the application of the specific ex situ technology is becoming more and more popular. Lower molecular weight contaminants and this kind of pollutant are very useful for the degradation by this method. Figure 11 ex situ solid phase system bioremediation by biopile is shown here. Now, windrow, this is the fourth one. Windrows are an ex situ bioremediation approach that increase the degrading activities of the native and or transient hydrocarbon classical bacteria existing in polluted soil by the periodically turning piled polluted material. The bioremediation rate which can be achieved through biotransformation, mineralization and assimilation can be accelerated by turning polluted soil on a regular basis, adding water and ensuring that nutrient, microbial degradative activities and pollutants are distributing uniformly. Next one is the slurry phase system in which we studied bioreactors. Degradation in a bioreactor is generally greater than in situ because the environment is more controllable and predictable. A bioreactor is a vessel used to transform raw materials through a sequence of the biological reaction into a specified product. The following are the many bioreactor operation modes. First one is fed batch, second one is batch, third one is continuous, fourth one is sequencing batch and fifth one is multi-stage bioreactor. Capital expenditure and market economic are the primary determinants of the operational mode selection. Bioreactor based bioremediation has several benefits, one of which is its excellence control over bioprocess conditions such as pH, aeration rate, temperature, agitation, substrate, inoculum concentration and substrates. Additionally, it is effective in treating soil and water contaminant by volatile organic compounds VOCs such as BTEX, toluene, ethylene benzene, benzene, xylene, etc. This is the bioreactor which is used for ex situ slurry phase system bioremediation. Next one is the mechanism of degradation by microbes. There are two types of enzymatic process involved in the bioremediation. First one is major type of reaction and second one is minor type of reaction. During major type of reaction, specifically involve oxidation, carboxylation, in which the carboxyl group is replaced with an H atom or OH group. Hydrolysis, which involve the addition of water to a molecule accompanied by cleavage of the molecule into two species. Substitution, in which one group of atom is replaced by another, such as OH for chloride. Elimination, whereby atoms or group of atoms are removed from injected carbon atom which remain joined by a double bond. Reduction, dehalogenation, demethylation, deamination. Condensation, in which two smaller molecules are joined to produce a large one. Conversion of one isomers of a compound to another with a same molecular formula but different structure of conjugated ring cleavage. Second type of reaction is minor types of reaction. In minor types of reaction, organic molecule leaving the main structure intact. First one is fragmentation, in which a complex organic structure in such a way that the fragments could be reassembled to yield the original structure. And second one is the complete mineralization, which in the transformation of the organic molecule to mineral forms. One example of describe all the three types 2,6-dichlorobenzonitrile. This is the figure in which various types of reactions are shown here, by which a toxic pollutant is converted into non-toxic or less toxic pollutant or product. Now, conclusion. Bioremediation is a natural process and therefore perceived by the public. Bioremediation is useful for the complete destruction of a wide variety of contaminants. Instead of transferring contaminants from one environment medium to another, for example, from land to water or air, the complete destruction of the large pollutants is possible. Bioremediation can often be carried out one side, often without causing a major disruption of normal activities. Bioremediation can prove less expensive than other technology that are used for the cleanup of hazardous waste. Now, summary. Detoxification of pollutants by bioremediation approach are varied and have been successful in cleanup of locations that have been contaminated by various pollutants. Since microorganisms are essential to bioremediation, information about their diversity, abundance and community structure in contaminated by monitoring other environmental factors that may hinder microbial activity and ensuring that they remain within an optimal range. Characterizing the site is the first step towards the successful bioremediation, since it helps determine the most appropriate and practical bioremediation process in an ex situ bioremediation. Because of the added expenses associated with the excavation and shipping, ex situ bioremediation are typically more costly, however, than can be applied to a variety of contaminants in a regulated way. In situ approach, on the other hand, do not additional costs related to excavation. Nevertheless, certain in situ method of the bioremediation may be ineffective due to the high cost of equipment installation on site and inability to properly visualization and manage the subsurface of a pollutant site. As a result, it does not seem that the primary consideration when choosing the bioremediation method for the contaminated site is the expenses of cleanup. This is the reference regarding this. Now, topic to be covered in the next lecture is genetically modified microorganism, in which we learn about the tool and the techniques to prepare genetic manipulated or modified microorganism, and the knowledge about the various genetically modified microorganism, their assessment of the safety and human and environment. Thank you. Myself Sabiha Naz, Assistant Professor, Department of Biotechnology and Microbiology, Bhilai Mahila Mahavidyalaya. Thank you so much.